Acute lymphoblastic leukemia, or ALL, is the most common form of childhood cancer. While the survival rate for ALL is high, some subtypes of the disease have a dismal prognosis. In 2009, St. Jude Children's Research Hospital scientists found a completely new and deadly subtype of this leukemia that arises from early T-cell precursors. The discovery was early T-cell progenitor ALL, an uncommon and very high-risk cancer. Most children with this subtype of leukemia, also called ETP ALL, don't respond well to treatment and typically relapse. At the time, researchers had not identified a genetic change common to all patients with this type of leukemia. A year later, with the launch of the Pediatric Cancer Genome Project, a collaboration between St. Jude and Washington University School of Medicine, researchers sought to understand the genetic basis of ETP ALL.
The Genome Project has given us the ability to identify all of the genetic changes in a cancer cell, or indeed in a normal cell as well. And because we hadn't found the genetic cause of this leukemia from other approaches, we thought, why not use this type of leukemia to fully understand a very aggressive subtype of leukemia.
One of the key things to understand is what's the difference, in the genome of these particular tumors. Are there additional genes that are mutated or pathways that are disrupted? And if so, can those be used as sort of genetic signatures to identify these kids very early on and then use that information to better direct their treatment?
Scientists identified 12 cases of ETP ALL that were treated at St. Jude. They then comprehensively sequenced the tumor cells and also the matched DNA from the same individuals with ETP ALL. Apart from finding these pathways commonly mutated, some of the pathways were also commonly mutated in other forms of leukemia that are different.
So that led us to suspect that this form of ALL may be quite unique and quite distinct from other subtypes of ALL. That is, that it may genetically at least be much more similar to an acute myeloid leukemia than an ALL. One of the reasons that that's important is that the treatments that we use for the different types of leukemia are quite different. In particular, different drugs are used to treat AML than are used to treat ALL. So one of the important clinical implications of this work is that we might be able to use some of the drugs that we currently use to treat AML to treat this aggressive subtype of ALL.
In the ETP ALLs, we found novel mutations that we didn't see in the other forms of ALL. These are unique to this particular subtype of the cancer. And so that gives us some new targets for therapies. It also sort of right out of the box gives us a better way to perhaps use a genetic test, a genetic diagnostic that helps better identify these particular patients and earmark them for much more aggressive treatment very early on.
When the experiment worked as we had thought it would work, there's such a joy and an excitement, because we're not just finding from the statistical point this gene is important, but functionally being able to test and verify and actually point it to a direction of where the potential new therapeutic treatment could be useful for these subgroup patients is truly rewarding.